Yes, yes.
So good morning, one and all. I, Dr. Meena Patilkar, on behalf of Youth Committee of HS Somaya College, Somaya Center for Experiential Learning and Kishore Bharati, welcome all the delegates from different parts of the country for the 2 days national meet. But as it is our Somaya campus tradition, we begin the program with the campus prayer. So may I now request all to rise for the campus prayer.
पूर्णमादाय पूर्णमेवावशिष्यते ईशावास्यमिदं सर्वं यत्किञ्च जगत्यां जगत् तेन यत्तेन भुङ्जीवा मा विदत्तस्य शिक्षणम् मूकं करोति वाचालं पङ्गुं लङ्घयते यत्कृपातमहं वन्दे परमानन्दमाधवम् ॐ प्रणो देवी सरस्वती वागीभिर्वाजिनीवती ीनामविद्यवती ॐ चोदयति सूनृदानां केतन्ती सुमतीनां यज्ञं दधे सरस्वती ॐ असतो मा सत्समय तमसो मा ज्योतिर्गमय मृत्योर्मा अमृतं गमय ॐ सहनावतु सहनौ भुनक्तु सह वीर्यं करवावहै तेजस्विनावधीतमस्तु मा विद्विषावहै सर्वे सुखिनः सन्तु सर्वे सन्तु निरामयाः सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखमाप्नुयात् ॐ शान्ति
Thank you. Now may I request all to be seated.
आपने ज्वाइन किया बिग ब्लू बटन पे कौन है? उसको बुलाओ इधर मैम आपका मीटिंग
I now invite Dr. Smita Subrase, Dean Academics, to welcome the gathering.
बीबीपी वाला कहाँ है?
YouTube, he is handling Big Blue Button.
A very good morning to one and all. All the esteemed guests and speakers from you and Kishore Bharati and dear participating faculty and students. On behalf of KG Somaya College of Science and Commerce, our principal Dr. Pragna Prabhu, and the entire fraternity.
मीना
I
मैम
extend my
कौन सा वाला
heartfelt welcome to all of you.
कौन सा
The
वाला
name of this
क्यूब वर्कशॉप
is CUBE, which is collaborative understanding of
ये क्या
biological
क्यूब
education.
फर्स्ट हमाया
It encompasses the two extremes of Bloom's taxonomy. One is collaboration, which I think is essential for creation, and the second one is understanding, which is the lowest level of the Bloom's taxonomy. The entire program is designed to enrich the student and the teachers' journey from understanding to the collaboration, that is the creation. Attitude
वो
to
बीबीपी चैट
attitude.
मॉडरेटर है
It is and will continue
ब्लू बटन
with an opening for all the young generation. And I think this is something, some platform where you can raise some fundamental questions and also seek the answers for that. I personally and on behalf of my college, K.J. Somaiya College of Science and Commerce, I thank for giving this opportunity for holding this today's national event in our college. I once again welcome to one and all present over here. Thank you.
Thank you, madam. You, as you all understand, collaboratively understanding biology education. This is the brainchild of none other than Professor Arvind and Professor Nagarjun. So may I now invite both of them on the dais to address the gathering.
इसका वेबकैम नहीं कवर कर रहा है ना उतने लेवल दिस वेबकैम इज कवरिंग तो इसको एलिवेट करना पड़ेगा तो जब बोर्ड पे आएंगे तो एक ही बात
Okay. Yeah.
उसको एलिवेट करना
What is the process to all of us?
तो इसको बुक्स रख के उसको ऊपर रखना पड़ता है और उसको मैं यहाँ भेज चुका हूँ बिब्लियोटेक चैट मॉडरेटर
To introduce you,
वो क्वेश्चंस वहाँ से उठा के राहुल को देना
my talk,
बस ये जानना चाहती हूँ कि बीबीपी क्या शो है
the cotting, that's what I
यहाँ पे
asked you.
इसलिए
I think the cotting, when he was in first year BSc or something, yes, in Acharya Narendra College, Delhi, 10 years ago. 10 years ago, and we have people who are whom we taught when they were in 7th standard. Abhijit, are you there? Abhijit, 7th standard. 7th standard. As
I have shared the link, please.
maybe we didn't take permission to catch an infant like that.
He's our host.
He is our host today. So, Rahul will be presenting some introduction, giving an introduction, and we have Meena here, who is an expert on science education, and she will be giving her viewpoint. She has been called to 2010. So all these things will be coming in a few minutes before you. So I think we have created problem for them like Rahul, Meena, and
इस तरफ chair लेके आओ।
that's how
You can check, you can
we have created some solutions and that problems being solved with creating more problems out of this, and we have people from all over the country. So that's what do you do?
Well, I mean, I sort of got hold of him after his retirement. So I did not let him go away because I really liked the conversational style in which he teaches biology. I mean, actually, he never stood in the classroom and actually delivered a lecture in Sophia College for so many years. So the conversation style, not chatShaala but a chatShaala. So we are actually seeking, I mean, as experimenters who are doing science education research, we are trying to find out alternatives. What actually would engage people? Okay. So one thing that engages people is chat, conversations. So he is our master. So one thing that he does, of course, is that he doesn't accept anything that if somebody says some fact, even if it is from textbook or even if it is from a journal, he'll say, how do you know that? You ask those questions, like how would somebody else come to know about it? Like Rahul does research in a very prestigious institute in the country, and we can ask him also the questions like how you have come to this conclusion. Well, with the same data, for example, would have given you something else. So this is the question. Science requires a lot of question. So the questioning has to become a habit. In fact, some of us may remember in earlier group meetings, the school students like Abhijit used to sit in the front of the university classroom, and some of our graduate students used to come and give a talk about Drosophila, Mozilla, Earthworm, and whatever they are doing. And then that small 7th standard, 8th standard student used to stand up and ask in the auditorium very courageously, do you have evidence for what you say? So this is an important STEM habit, or a science habit, an academic habit that every one of us should have. And we start asking these questions. Why? Because textbook is our Bible. We believe whatever is in that textbook, and then we take everything that is the teacher is asking, because that is what comes in the exams. We have to go beyond that, because we have to be honest to ourselves, and we have to be honest to the practice of science. And so we have to ask these questions of, how do you come to? Suppose somebody says, this one is 6 micrometers in diameter. So what do you mean by 6 micrometers in diameter? How does it relate to what you see? Can you see 6 diameters of 6 microns under a microscope, or can you see it actually with the naked eye? All these are experiential aspects which Ashutosh's unit here in Somaya College is looking at experiential aspects of learning. So it's a wonderful occasion. Thank you so much for hosting us for this important meeting, and we certainly want, because we don't want anything 1 day, 2 days, 4 days kind of thing, because now thankfully we have to do that here. So particularly all the Somaya students here, can you raise your hand? Okay, now, so look at this. Meena is also raising hand because she's an alumnus from Somaya. She did marine biology. Marine biology, right?
MSc.
MSc marine biology from this very institute. And so this is these are the people who are going to continue this cube lab here. Okay, so that's the kind of thing. So enjoy conversations and continue these conversations in the evening chatShaala from 7:30 to 9:30. We don't stop them even on a holiday or Sunday, Monday, whatever it is. Even if 5 people come, we have a conversation. If 50 people come, we'll celebrate even more. So we'll continue this conversation. And you want to add it?
No, I think I will invite Rahul to come and present a few slides to explain what he's experiencing.
Yeah, good.
Thank you, Professor Arnab and Professor Nagarjun, for sharing with us the story of the inception of Cube and giving us an overview about Cube. So thank you, sir. And now I request Ashutosh More from Somaya Center for Experiential Learning to take it further.
So as we move on to calling our guest speakers for the day, I'm really happy to know that we are calling here Rahul Kushwaha. And I say this because Kushwaha's story is like he has been a cubist to begin with. Okay, and like all those who raised their hands right now, and even the non-Somaya cubists, other cubists, Kushwaha joined the program as a cubist, as a young cubist, and he had Kushwaha over here, Rahul, is the product that we have of the cube culture, right? So when Jane said that he's pursuing his PhD at a prestigious institute, he is one of the products of what we had through the cube culture. So I somewhere feel he's the best person when it comes to talking about cube, right? And so I'm happy to call upon Rahul, and we will be talking about, I think Rahul, you'll be touching upon the gully cricket model, which I believe all of us should be aware of how cube works. So yeah, I invite Rahul to come over and have his talk.
Thank you, everyone. So I'll not be standing in a corner because that's not what you taught me. So it's both a pleasure and a privilege to be among all of you. I'm always delighted to see cubists, and it's happening for the first time after the COVID. So it's also a very auspicious occasion on that front. And as very rightly pointed out, I am a product of Cube, and I'm very proud to say that, because few lines that always like that are embedded and you can say stuck in my mind is what Arnan and Gian always used to say throughout this 1 decade of me being in Cube. One thing is that both devil and divine lies in the details. We all started from there because yesterday only I was discussing my own journey with some of the cubists. I was telling that 10 years ago, so currently I'm doing a PhD in neurosciences, but 10 years ago I was standing in front of Arnan and I was trying to draw a neuron, how a neuron is there in our hand and how we get reflexes when we touch something hot, or So I remember I drew from the fingertip to the spinal cord a series of 4 neurons connected in a haphazard manner. And then Arunan was back in ANDC in my first year of BSc. I was so nervous and he told me, oh, so great. So we came up with a model that Rahul has given us. Maybe all our textbooks are wrong. So let's just discuss this. So at that very moment, I was not encouraged, I was not discouraged, but also my mistakes were celebrated. And today, after all these years, I know a lot about neurons and I still know very little about neurons, and I'm fascinated to know more and more. So this is what Cube has done to me. This is how it has changed me, and I think it will do the same for all of you over the years. Now, as I told you, it's both a pleasure and privilege to be back in these offline meetings. And what today I'm going to talk about is, so we all know Cube, and I want to welcome you all to the national meeting. So CUBE stands for Collaboratively Understanding Biology Education. If you take any word out, it doesn't make any sense, right? So it all starts with collaboration. Hence, you can understand what are we understanding is biology, and it's all related to education. And another line that's set in my head is what always GNNR says, that to cover the syllabus, because syllabus is something we all are always worried about, right? If I ask any one of you at the moment, so what is the next big thing you are worried about, is the midterm exam or this or that, and the syllabus about it. So they always used to say that to cover the syllabus, you first have to uncover it. So let's uncover the syllabus and understand what we have to cover and why we need to cover that. Mostly in schools, sadly, what they do is they technically cover the syllabus. Take a bed sheet and cover it, so no one just sneak peeks inside it and ask questions. Okay, so we remember some answers. So let's go further. Basically, we talked about gully cricket. Why we're talking about gully cricket is because if I ask you one thing that's very famous in India and unites everyone together, regardless of their regionality, religion, caste, creed, their status, is cricket, right? We all enjoy cricket a lot, and especially everyone plays cricket. We don't have stadiums in every gully, but we have cricketers in every gully in our country, right? So the whole idea is good scientists and good science takes inspiration from society, and this is one inspiration that we in Cube took from society, is to make science like a gully cricket model. So what do we mean by gully cricket model is, if we go forward, is so we need to create spectators for science. Like if you see this picture, see that a young boy is playing, but he's not the only one enjoying the game, right? You can see a lot of people amidst whatever they were doing in their day. They're all gathered and they're all enjoying. Now there are certain things to learn from this. If we do, let's go back before. Let's go back. If we try to understand what's happening here is, so this boy who's playing clearly is enjoying because he's in the game, right? He's dictating the game. But the others around are also able to enjoy because they also know the rules of the game. They know what's happening. So when Sachin Tendulkar,
the mic is not working.
It's on. Yeah. So when Sachin Tendulkar hits for a six, we all enjoy because we know it's a six, right? We know what it means to the game. This is what we have to create for science in India. People should know what it means to do science, and people should not just distance themselves. So like we all do science with taxpayers' money. We are not generating our own money and then doing a lot of science in the country. But the taxpayers know nothing about what we are doing behind the closed doors, using crores and crores to make machines and do things in a sophisticated manner. So that's where we have to open those doors and let people come in, know the rules of the game, and cheer and become the spectators of science. That's what you wanted to do. So let's go further. Let's talk about some great cricketers. Oh, okay. So how many of you know this person? How many of you know this person? I said let's talk about some great cricketers. So, anyone would like to take a hint? Who's he? Looks like the walls of Somaya only, right? The walls of any institution. So who's this person standing in front of the wall of any XYZ institution? You might not be knowing him, but if you go forward, oh yes, that was Arunan. Maybe in a parallel universe 50 years ago. But you all know this person, right? You all know this? Who's he? Anyone? Yes. Perfect. So he is Venki, who got Nobel in 2009. So he's by training a physicist who worked on ribosomes to figure out the structure of ribosomes. Ribosomes also have a unique story in my educational life, but I'll tell you later sometime. So he worked on ribosomes, figured out the structure, but he got a Nobel in chemistry, right? So it proves that basically what he did was science. You cannot cordon off or make boundaries in science by introducing names like physics, chemistry, biology, right? Let's go one photo ahead. How many of you know him?
So Rahul,
Exactly. So this is where we got the idea.
Rahul, we have the participants online also. You can address them. They'll be also respond to your messages in the comment box.
Okay, great. So we have participants online. Please do respond in the chat box or message box. We'll be taking care of your questions and answers as well. So yes, we all know Virat Kohli, and that's where our idea came from. That everyone knows Virat Kohli, but not everyone knows Venki. So there's some issue in that. Let's learn what's the problem. So the issue is cricket is so famous, and there are some reasons why cricket is so famous in the country. And if you go forward, can Indian science learn from Indian street cricket? So science should learn from each and everything, right? So look at this picture. What's happening? What's happening here? Yeah. Okay, let's go to one game, one picture again. What's happening? I see some very happy, elated faces. I also see an umpire who's just like, I think they're just from the same society or something. So, but all of them are very happy because they know how to play this game of cricket. Hence, they know how to enjoy it. So, knowing the rules of the game enables you a lot. There are no complaints. You can just play the game with whatever you had. I used to remember playing. I used to carry a lunchbox to school, and we used to wrap rotis and parathas in aluminium foil. Then after you have eaten the paratha, you just make the foil into a ball. You have the lunchbox that becomes the bag. So that innovation just comes instantly, right? We all have done it. No one came and taught us or gave us a protocol. See, you can use this as a ball and this as a bat. It just comes on its own. So even if there is lack of facility, we can do that. Improvisations, wickets. We don't have stumps in India, but everyone plays cricket just by breaks. Not just breaks. If you see, everyone. So there is, yes. So everyone can play cricket. It's for regardless of gender, regardless of your economic status, regardless of where you are, regardless of. And look at this young Virat. I think it's Virat Kohli only. Again, just like in a parallel universe. And look at the expression. Can you make out any difference between this cricketer and an actual cricketer who's padding up with all these branded gears and playing cricket? No, right? Because what they share here is the same joy and passion and the emotion for the game, right? So this is basically what we as scientists can learn from the gully cricket model. So it's the same feel, and here comes collaboration. Why do I call collaboration? So look at one field is where the game is ongoing. Some people are waiting for their turn. So it can be their pitch as well. There is another pitch that's already in the same ground there are more than one pitch and the game is happening parallelly. So that's called collaboration, where we can work with each other. Lack of space, lack of resources does not matter. Let's just squeeze in, let's just adjust. So for gully cricket we adjust to any length, we go to any length and we adjust because we get a sense of joy, a sense of elation in the game. So why can't we do the same for science? Why is science alienated? So it's the same rule. Science also have same set of rules, whether it's done by Venki or it's done by Arunan or it's done by Abhijit. It has the same rules to go forward. So now if I come back and if I ask, why do we know Virat Kohli and why everyone knows Virat Kohli? The answer is because, let's go to the next slide. Yeah, we talked about Venki already. Now we know Venki as well, right? I hope that when you will go back from here now, you know who's Venki, right? Okay. So the idea is let's implement this gully cricket model on science. How are we going to do this? So first thing is let's start with our own pitch. So this was the first pitch that we kind of made. This is the cube lab at HBCSE where I also spent a few summers, and we have a similar kind of lab here at Sophia. So that's another pitch we have made ready. So it's the same model. So what happens when suppose you are playing on a pitch and next day some construction workers come from the municipal corporation, take your pitch away, then you go to another pitch and you start playing. But the game never stops, right? Whether you have to play it on the roof, in the gully, you don't stop playing. So that's what the idea is. And these are very happy snippets from the causerie sessions. There were offline causeries that used to happen in Cube Lab. And I'm sure here also you will be doing offline causeries, you will be meeting, you will be talking, and like on the way when we are talking, why should we just talk about gossips? Why can't it be scientific gossips? Or why can't it be about a model system that we have at home and we are trying to ask something? So causerie really transformed, this talking really transformed the Cube chatShaalas, and as we go forward, I wanted to, so another thing which is important is just like the score cards in cricket. So every match is a scorecard. We always, because the rule of the game include how many runs you scored, who did what, who was the man of the match, who was the man of the series. So even if it's gully cricket, we are always happy that, oh, I scored more than you. So the same concept is here. I recorded more than you. I recorded thrice a week. I have written about C. elegans. What have you written about? So I have written about Drosophila. So we started recording these things on the Meta Studio, and these will always be there. This is, I think, the first screenshot of the Cube chatShaala, the online version of the causerie, which started on 21st March 2020. It was because amid the pandemic, the offline causeries could not happen. So again, whatever happens, the game never stops, right? Remember, that was the model of the gully cricket. So the game didn't stop. We took our pitch online and we started hitting sixes and fours there. And currently we have grown, and these are all the Cube home lab centers in India and even outside India, and we are still going strong, and that's a good thing. So our gully cricket is going, literally it's going in each and every gully, and that's the plan, right? You cannot play gully cricket in just one gully of the country. So it's been 1200 plus, if I'm right. Kiran just told me in the morning, today is the 1232nd day of Cube continuously playing this game on our own pitches everywhere, right? So this is some of which we will talk about in today's presentation. So just some statistics to tell you that people join over the online Cube chatShaalas, and just the numbers. The Cube culture is growing everywhere, and what is happening in these? So we understood that we took it to each variant corner, but what is the game that we are playing? So talking about what we are playing is this. So science can be done in biology. We use model systems, right? We call them model systems because there are certain questions that we cannot address if we do not have a model system to answer that for us. So for example, if we are talking about very simple things like activity and inactivity patterns, or something like culturing of an organism. So we need to culture an organism first to ask the question, right? Exactly, just like we go, we get a space, we prepare, we put bricks, we make a wicket, we make a bat and things. So these are our equipments with which we are going to do the science. So people started in cube culturing these small flies called fruit flies. I'm sure that all of you know what are fruit flies, right? So they are the tiny flies that keep hovering around fruits, and this is one video that showed you. So these are the ones that keep hover around fruits. So this is, I think, on a fruit pulp. They are also called vinegar flies for the reason because they are very heavily attracted towards the smell of vinegar. And we can just stop the video, and then you can also find them on tomatoes and some pictures forward. This is how cubists started trapping fruit flies at their home. So you give them a bait, they come, and then you trap it, and then your game starts, right? So you have to trap and you have to culture. So we started with what's actually first written in the textbooks. Like Arman said, we always go to textbooks like it's something very sacred, and whatever it tells is the only truth in the world, right? But here at Cube we question, is that really so? Is that the only truth in the world, or we can do something different? Because this standard corn meal agar media for fruit flies is used in every lab. Most of the labs use this. But what cubists thought is, can we do something of our own? Is this the only way to culture fruit flies? And see, this is a question that is uncovering the syllabus, right? Most of the schools, the teachers may not like this question. See, I'm telling you, this is how you have to culture fruit flies. But if you ask, can we culture it differently? So that's a question that we encourage here at Cube. So we came up with, as Cubists, a home lab based media, which is called the TRSV media, which has tomato, rava, sugar, and vinegar. Remember, that's why the vinegar, they are vinegar flies. They are attracted to vinegar. So, and they look exactly. So what should be a good media like? A good media should have a composition. It should be solidifying so that Drosophila can lay eggs on it. So this is what a TRSV media looks like. It's quite good. This is how you make it. So we also made a recipe of our own. You add water, all the ingredients, you cook it, and it's done. Now, going back to Seymour Benzer's time, who do you think gave Seymour Benzer a recipe to culture fruit flies? No one, right? Maybe he figured out a way. And Seymour Benzer used to keep flies everywhere, in his office, at the windows, so that they get proper lighting, in his room itself where he was living. So he was very passionate about culturing. He figured out way, and maybe that's how he also figured out how to culture fruit flies, like our cubists figured out how to culture fruit flies at their homes, right? And not just, so is this a video? Yes, so this is a video of home lab cultured fruit flies. So you can see labeling again. So when we are doing science, we have to be meticulous about what are we labeling. Our labels should not be wrong, because labels tell a lot about what you are doing. So in the labeling, you can see it's successfully cultured because you can see a lot of larvae and pupae in the bottle, which means the flies are happy. That's why they are laying eggs. That's why the the eggs are also developing and they are becoming larvae, because they are getting nutrition. So that's a successful result of how to culture fruit flies using our own medium. Yeah. Then comes identification. Cubists were also looking at how to ID fruit fly. We came up with many things, many questions, how to identify. We take literature, and one of the ways that we can identify Drosophila melanogaster, which is the fruit fly that mostly is used in the labs, through their wings. Now, in the wings, there are breaks in their postal ring here. So you can see these 3 breaks, which are the characteristics of this species, and if you see these 3 breaks, you can identify that this is Drosophila melanogaster. Now, this is just an answer. Now, to do this, you will come across many issues. One issue is how to image the wings, how to see the wings and prove everyone that this is Drosophila melanogaster. Then Cubist from biology takes a leap into other arenas as well, because we learn. Imaging is one issue. Not everyone has a microscope at home. So should we stop doing science? Just like not everyone has a wicket at home or bat at home, do they stop playing gully cricket? No. So that's where we don't also stop. So what we can do, this is what I like this particular slide, because they say it's Aswati's phone, so it's still here with us. So this is Aswati's phone. We have kept it from a very long time. What she came up with is, you can put a light source. What is basically a microscope doing? It's giving you a light source and a magnified image, right? So in the phones, you have the ability to zoom in or go closer and take clear pictures. You just need a illuminated subject to image, right? So you keep the wing specimen here, you illuminate it from the bottom, and then also put a transparent slip, and then you image from top. Now, will it solve the problem? Will it be a good home-made jugaad for us, just like the TSRB media? Yes, it looks like it is. So we can get very, very clear pictures like we get in textbooks, and we can clearly see the coastal breaks and talk about, yes, this is a way to identify Drosophila now. Along the way comes improvisation always. When you are doing home-based methods, even in labs, not just home-based methods, even in the labs in the countries, when people do science, not every time sophistication helps. Sometimes you have to do improvisation, because that's what makes you a scientist. You come up with innovative ideas and do some jugaad.
So this is their phone microscope. You can see the torch and the wing is kept on the transparent sheet.
Yes, so this is the phone microscope, and you can see, so this is a torch, a wing, and I think you just need a sellotape to put it in place, and then you can image it, right?
These are the pictures.
So these are the images. Great, we did great. The Cubists did great. They could now identify Drosophila. And so a very simple model system like fly, which was captured from the wild, is now allowing us to do real research in our homes, right? That's where Cube become infectious. So one person does it, and then another person sitting in the room thinks, oh, if he can do this, why can't I do this? And it looks very simple, cost nothing, let me also do this. So that's where we are taking from that model of gully cricket. So if I am playing, the other person will also, come and start playing with me, because it's fun, we are doing it together, and it costs almost nothing. Now, you must be thinking why we are showing you potatoes. This is not the menu for the lunch, let me tell you. Do you mind potatoes? But let me take you back to the time of Robert Koch. You all know Koch postulates. Those who study microbiology must be knowing about Koch postulates. Koch was the first to say that there are some agents, very, very small microbial agents, which are responsible for causing diseases and transmitting diseases from one person to another. They're called bacteria. So if they are really there, how can we show that they are there? How should we culture them? Again, see, culturing is a problem, right? So to culture a bacteria, let's say, which you cannot see, which is very, very small, let's come to logic. Let's ask what it needs to survive. So it needs nutrition, right? It needs a bed where it can lie, just like we wanted a bed for Drosophila larvae to grow and eat. So how about using a boiled potato, which you cut into halves, and it's sterilized, and if you allow bacteria to grow on it, if you incubate and make bacteria grow in it, it will work like the media, which is the artificial media, agar-based media, which is used in labs to culture bacteria. So you can even do microbiology at home. This is exactly how Koch did. So Cubists took inspirations from Seymour Benzer. Cubists also took inspiration from Robert Koch, and you can see it works really well, right? Very, very beautifully, you can see some bacterial colonies here, exactly like we see on artificial media in labs where people culture bacteria. And yeah, this is amazing, a very healthy bacterial lawn. And so this is the good thing about collaboration. One person does it, and he figures out, oh, I can culture bacteria at my house. How about you try it? And we all have potatoes at house, right? We all have some broomsticks to make wickets, bricks to make wickets. We all have potatoes at home, and let's do it. So everyone started doing, and not just bacteria. Then you go one step further and ask. You don't stop there, because now you are into the mode of being a scientist, and there is no end to being a scientist. So you ask, okay, can I also culture other things like nematodes? So these are small round worms, C. elegans, and yes, you can do. For them, you just also have to give. This is just to keep them, I think, bounded. So these soils will have the nematodes, and this will provide a lawn to them to grow and culture. And in the next pictures, you will see if it is successful. So these are some setups that are done by
so we have
cubists.
the video for this.
Some cubists, and I think this is the video where you will see whether this improvisation in Jugaad was successful or not. And it was brilliantly successful, right? You can see that people could culture nematodes, bacteria, fruit flies. All these are model organisms to do actual fundamental real science at home, right? Great. Okay, so let's just see one of the cultured. So from the culture, which are on potato slice, in this video they're just imaging it under a fold scope. So you just take some of them, spread it on the slide, seal it, and then just put it under the fold scope and see. Let's see what it looks like.
Ooh,
Wow.
nice.
Right? It was amazing. It's just like, so fold scope is another handy tool that you can use. It's a pocket microscope, and it's amazing. So all these enables everyone to do science and to feel that science is as easy going and as fun as a gully cricket could be. So that's our model, and we hope it's an infectious model. We don't just stay like get we don't just stop at animal systems or microbial systems. We can also do plant studies. This is something we all have been doing at our homes. I'm not telling you something which is very, very unique and you're seeing for the first time. But okay, moong, dal, and all these legumes always grow, sprout. We sprout it, we eat it, we don't think about it. But can we use it to answer some questions in science, some very fundamental science, evidence-based research? Can we do that? Yes, surely we can. So here you can see, in a very systematic manner, cubists have seen the development from a seed to this stage. And what it teaches us is that recording is very important in science, right? Suppose I just show you this and I show you this, the starting and the end picture, and I don't show you anything in between. That's what happens to us most of the time. So my mother used to put sprouts before one night, and next morning I'll just wake up and eat it and go to school. But I'll never think what happens in between. If you start asking what happens in between, that is exactly what science is, right? Because then you will go and systematically record. You see that it has a rate of development. So basically what you are figuring out here is evidence-based plant germination, plant development. So you're using plant as a system to address questions of development. So from just curiosity-driven things in your day-to-day life, you can go to doing very cut-edge research at your own homes, correct? So feels good, right? Looks like, yeah, we all can do it. That's what Cubists did over these years. And so here, what Cube has done is made this gully cricket model possible. Many of us has just taken it up, including me myself. I remember a night when it was a 1 day's work that allowed me to realize that model systems, in the plethora of model systems, Drosophila is my best model system. And Arunan is the witness here for that day. I just sat over a day and started seeing a pupa, a larvae, third instar larvae becoming a pupa, and that pupa getting colored over the time. And I just recorded it, and I saw that in a span of 6 to 8 hours, a white pupa becomes a darkly colored yellow orange pupa, right? And that was amazing. So all of you can find your model systems like this. Once you find it, there's no looking back. And I would like to So this is a featured article in India Bioscience undergraduates in lockdown. So even lockdown could not stop. Let me tell you, lockdown may have stopped gully cricket. So here we are learning from the model, but we are going one step ahead. But lockdown could not stop the cube chatShaalas. And in lockdown, there was nothing to actually do for the world. We kept learning, discussing, talking about science, doing these experiments at home, and felt good. So I'm very happy to be presenting this, and I'm glad that I did it. And thank you, Kiran, for making this presentation and which allowed me to talk about Cube so much. And I would like to thank Arundan, GN, and everyone to invite me here. It's amazing, and I'm looking forward for a great 2-day session. So this is all about Cube, and cheers to us. And let's open the meetings. Thank you so much, everyone.
Okay, so thanks Rahul, thanks for this wonderful talk. So I guess the last part where he had this flashing on the screen that gully cricket model is possible, home labs are possible. I think that is the takeaway message from the entire talk that he had, because he gave us enough examples of how that is possible. At the start, he told us how in cricket we do that. We do not have excuses. We want to play cricket as kids. We have all played cricket, and we don't have excuses. We don't have stumps. We somehow get anything and make stumps out of sleepers, right, or something like that. And then he gave enough examples of how we could do that in science. And that last part, I believe, is the takeaway message, that gully cricket model is something that we can replicate in science, and home labs are certainly possible. And cubists over here, all over the nation, are doing active research in their home labs. So thanks, Rahul, for this presentation. So I would now like to call upon our next speaker, Ms. Meena Kharatmal. And so let me just give you a little introduction of her before I call her upon stage. So I'll begin with my story of how I know her. So I joined for the CUBE program as a junior research fellow in 2012 at Homi Bhabha Center for Science Education. So that is the time that I met Meena. She was, I saw her as a active researcher working there. She was a scientific staff. She has been a scientific staff at Homi Bhabha Center for many years now. And that time I saw her as a PhD student, and her area, the area that she works in, has been in knowledge representation in biology education. And that's what she has been working all this while. And she had been very actively connected to the CUBE program, and I had joined over there as a junior research fellow working on the CUBE program. So that is how I got introduced to Meena. And as I said, her area has been knowledge representation in biology education, and she also has been very active in creating online resources that we use for CUBE, given examples like CUBE Chat or STEM games or STEM chat. So all these online resources that we use in CUBE, she has been involved in contributing in that. Apart from that, during the COVID, TFR had this initiative called as COVID GAM, okay, where they wanted to, so there were not many doubts that we had about COVID when it came, and what are vaccines and this and that. So for the general audience, TFR created this online resource where the most common questions, the frequently asked questions of those would be answered by scientists, and Meena had been actively involved even in creating that particular resource. Okay, and with the CUBE program, it is also that she has also been trying to do this, that CUBE as an intervention, how is it bringing about learning in students? Okay, so I have also been involved with her in working on this, as when we say CUBE as a model is what we use for students, what kind of learning is it bringing out in students? Okay, how is as CUBE as an intervention is working? So I think she'll talk in detail about that. So let me call upon stage Meena to have a talk.
Thank you, Ashutosh. So good morning, everybody. Let me handle the.
These are your slides.
But I'll be switching. Where is the here? ना ठीक है ठीक है ठीक है. It's okay. How do you start the slideshow?
Phone पे करें.
Okay. This will go right. Okay. Good morning, everybody. I think Rahul ended his innings with curiosity to frontier. Now it's my time to bat. So I'm going to take this forward from curiosity to frontier. Now, how many are you newly attached to Cube? I just want to know. How many are newbies in Cube? Okay, very few. ठीक है.
Debutants in Gully Cricket.
Yeah. so, so there is a small population or spectators who are new to join Cube, and the others are sort of already batting since so many years or months or days maybe. So I was asked to talk about Cube, then something sort of, even for the Somya College who would be curious to know what is Cube. So I have a slide presentation like an invitation to Cube. Suppose if there are new people who are curious to join Cube. So we wanted to present what is Cube, some kind of a philosophy behind this model, how does it work, and what is the sort of collaboration behind this model. So I'll give a short glimpse of about that model, and it's like an invitation to Cube. And as Arunan said, GN said, they are creating problems. They are creating problems for the students. So I have figured out one problem, and what is that problem? I'm going to also present. Okay, teachers. So I'm going to present my problem. What I have seen a problem in this whole Cube culture or Cube model. So let me begin.
If you want, I can show.
You can show. So this is a very favorite statement. Arunan likes it. Anybody read it? Yes, you please read. Read it aloud. Chalo jaldi. Read it.
Can we give
Anybody.
to the participants?
It should not be something
I don't want
we do to our students. It should be something we do in collaboration with our students.
I wanted you to read the first
We
statement.
missed the main thing.
Yeah.
Sophistication is required in the mind and not in the lab. It was said by Obey Siddiqui.
Yeah. So Obed Siddiqui is one of the pioneer scientists who sort of created this model of Drosophila, right? In TIFR, he set up a lab and he worked in a lot of depth, creating a frontier research area. Anyway, so what this statement mean is that if you're curious, if you want to do science, then you should. There is no end to imagination. First of all, that is a statement that we have to understand, and you should not think if there is a lab, I need facility, I need sophisticated instrument. No, sophistication is not in the lab. In this here, you say lab means not in the facility or big instruments, but it should be in your mind. So that sophistication has to come out. So that is what I think Cube tries to create. And of course, the second part was about the education, wherein the C, which stands for collaboration. So it has to be a collaboration between students, teachers, parents, and even citizens. So let's go ahead. So this is a very sort of, when I was creating a picture of where are the different labs. So this is just a snapshot showing how across the entire country we can see Cube labs. So of course the red dots that are shown are the various places that Cube labs are set up, and people have been already working in cubes and asking questions right from different parts of the country. So the main point here is that the program has a participatory base. So in science education research, there is a program called you have to learn or engage students using their own participation. So it takes care of it. Next slide. And here, I mean, see what I am trying to tell. Through each picture is the model.
model or philosophy behind cube model. So what is here, we wanted to show cube has a vertical integration of school and college students and teachers working collaboratively. So this is, whenever there used to be national meet, I'm sure after 2 days you're going to have a similar picture. So what is happening shown here is that there are teachers, there are scientists, and there are young students, there are undergraduate students, school students. So this, and behind the photograph you can see some canteen staff who used to be very much helpful to the students whenever they used to go and ask for banana or milk. So they used to happily provide all this. So this means there is no divide. This community has no divide or partition. So the vertical integration means right from school students you can engage up to scientists and undergraduates and postgraduates. So there's a mixed breed, you can see. Okay, next figure. So who can join Q? So this was a sort of a poster which Rahul showed you on the first lab. It was set up in HBCSE. Me, we, geek, everybody is welcome in the cube. So that's also one of the philosophical model. Next slide. So cube has no selection process. So in our center also, we have various programs wherein what they do is you have to apply for a program. Then there will be some fee, and then you have to pass some written test and all. And then only if you pass, you get some score, then you will be allowed to engage in that program. So something like that is not there. So it is all dissolved. This selection process is dissolved. What is the selection process criteria? If you are curious, if you want to engage, if you want to collaborate, you are welcome. So that's another model that we welcome in the cube. Where can cube be started? So there was a place, gully cricket. So where all we can start? Of course, you have college lab, you have school labs, but we have kitchens also, home labs. That also can be turned into even your backyard, your ground. Wherever you go, maybe to a forest. So you can just start within a window or so any constrained space is not required to be set up as a lab. Next. So what do we mean by cube for students? So you can see here, this is a busy day when there used to be causeries in Hoya Center. So you can see the students, sometimes there was a question and then the doubt was getting resolved by doing some causeries and drawings on the board and all. So they used to not just come here and talk and not just answer questions. Whatever was in their thoughts, you have to articulate. So articulation became a very sort of important affair in this causery. You have to speak, you have to articulate, you have to draw. Maybe writing was happening now it is happening in this medium of chatShaala. So there also you have to write, right? So next. So here I'm giving a glimpse of various model organisms that people, students are engaging in. That figure, go to previous slide. So here is a brief snapshot. I think Kiran used to work in cardamine. Then rotifer I used to like a lot. I used to also observe rotifers in the Pagelopos model. Then earthworm, snail, fruit fly, which Rahul has mentioned, and mosquito and hydra. Hydra was another popular and my favorite actually. Hydra has another story. With these model organisms which we see, with the day-to-day affairs, you can connect, as Rahul was also saying that, curiosity to a frontal level question. So what is this connection I wanted to make? You are not just doing any, observation-based studies and all, but you have a problem, right? You are observing Drosophila with a particular problem. What is that problem? Now for Rahul, maybe something, he wants to understand sleep pattern. Maybe for earthworm, you want to understand how can it, regenerate and all. So there is some question, right? Even within cube, we are saying, what is your question? What is your curiosity? And then you take a model and then try to, understand your question. So when we say from curiosity to frontier level research, what is the connection? That through through earthworm you can make connections to regeneration or spinal cord injury, which is a frontier level research area. So through Moina, I am sure you may have heard or done some studies on Moina, right? The water flea, right? Yes. Hmm. So that you can connect to epigenetics and studying hypoxia, which also has this problem of hypoxia has won a Nobel Prize recently, a few years back. So similarly, fruit fly, the studies on fruit fly has also won a Nobel Prize. So when students are engaged with these model organisms, what they feel, oh, they are also working in the area where scientists have received Nobel Prize. So that that thrill they get, okay, we are also doing some kind of frontier level research using with very simple level model organisms and which are there in your garden or in your surrounding. That is also very important. You are not doing any studies with any model organisms that you cannot acquire easily. So that is also one of the what you say important thing that we should understand, that it is in your locality surrounding that you are observing things and still connecting, learning biology and connecting into the various research areas. Next. Okay, next. These are all okay. So during such national meets, what we do is we engage students and ask them to draw some kind of flow chart or mind maps, wherein they have to make connection of what frontier areas that you are using, and through this, what is happening? This gives a context to connect to the curriculum. Right now, in colleges, in theories and all, you may not understand the chapter, or any typical chapter will not be Drosophila and sleep pattern. क्या होगा चैप्टर्स में? सेल एंड मॉलिक्यूलर बायोलॉजी, राइट? ऐसे सेल सिग्नलिंग, ऐसे चैप्टर्स के नाम है, राइट? बट वी हैव टू कनेक्ट जो भी कॉन्सेप्ट्स उस चैप्टर में है to what you are learning with. So we ask students during some workshops, why don't you make a map of that? Whatever concepts are, you may learn, read, understand, clarify through your model organism. So that was a nice thing, that this model organism, this study gives a context to connect to the curriculum. So very beautiful maps students have drawn. These are the actual maps drawn by undergraduate students like you all, and even school students. They have worked together and drawn these maps. I'm sure it will be there in the cube lab somewhere. Next. When such meets happen, when do they happen? Whenever there is a vacation time. So we used to have lots and lots of students and overflowing labs with the campus and all in all these vacations, like Diwali vacation, Ganpati vacation. Ganpati was a recent addition, so we took the opportunity. Okay, now 5 days to 5 days, chalo, aa jao cube mein. So Christmas vacation, summer vacation, winter vacation, whatever. And see, students used to not just come to the cube lab only during vacations. The spillover happened throughout the year. So once they are sort of infected, they don't want to leave the lab. So that was also one of the participatory model. We used to welcome everybody. If you are curious, if you want to continue your work, you are welcome. ऐसा नहीं कि ओके वैकेशन खत्म हो गया, वर्कशॉप खत्म हो गया, 9 से 5 और मंडे से फ्राइडे खत्म, ऐसा नहीं है. The work is to go on. I think it is still going on. Next. And then I'm sure this also you are aware of, how it connects not only to students and teachers, it also tries to connect with the citizen science program. And mango tree mapping is Arun's favorite. So, wherever he goes, he'll take pictures and he'll ask students to draw a map. And then what is the idea behind mapping? Understanding the morphology and flowering pattern, and, the pattern of when the fruits occur after flowering and all. So that pattern throughout the country he wants to just study, and then you can also attract citizens into this affair. So what you have to do, you have to just click photograph and then put some kind of coordinates and date and month. So this is also a form of data which you can collect and make some findings. Next. And okay, so
another point I wanted to make there was about nail regeneration. So whenever an event of election or something, comes into our state or country, so we get this ink mark now. I think आप लोगों ने भी voting किया होगा, right? So with that particular ink mark, you can study nail regeneration. So what you have to do is to take picture and then plot it around. Keep your finger on the graph paper and take some readings and observe how the nail regenerates. So that was also another context. Yeah, of course, these are the maps. The data is coming from various parts of the country. This was another exciting thing that Cube Movie Makers Club. It was started, and then the first context that was given for students to make videos on DNA extraction by Jugaad. Now we know that typical undergraduate what to this research centers sort of identify or observe DNA through sophisticated instruments like they run PCR, gel, and etc. But I think with Jugaad means easy. Whatever materials that are available in kitchen, you can still manage to get DNA extraction. So that kind of what you say context was given for students like a competition, and then they came up with different videos. I'm sure Cube will have those videos somewhere. So that is also another interesting thing that you can take up. Next. And I'm sure this is not new to you. So how does CubeNet work? Through instant messaging and this, I think the STEM chats or STEM habits is the program that you are now engaged with. So that is another way to create a network. Next. These are some glimpses of teachers. I want to point out we have been talking about school students. I think this is Abhijit, this is Gaurangi, this is Arif. This is also an interesting place. It's a school run by municipal corporation, and Arif has been very active and very exciting. He kept the Moina model organism alive in our lab whenever, and he was like, okay, Moina chahiye, I'll come whenever. So he was very much enthusiasm and he used to bring bucket full of water and lot of moinas, and he knew how to keep them survive. And of course, I know what moina is being studied here. And okay, so you know how does it look like? It's small, transparent, right? Next. Okay, so we'll go ahead and I want to. Okay, take it. Okay, so I want to just take a pause here and इसका mouse नहीं चलता है? नहीं, मैंने disconnect. These are some publications wherein we have tried to write something about Cube in at least English, Hindi, Marathi languages. So we wanted to have its reach to different places of the country. So through Eklavya we tried to publish it in Sandar. Through SSC board we tried to publish it in Marathi, so that a lot of people can read it. Because the purpose is that Cube should not be at one place. It should percolate inside the various places. So that was a motive behind writing this invitation based articles. And home lab, of course, we have written about it during pandemic. What we did is we interviewed few students and asked them what are their challenges, how are they trying to keep their model organisms alive during pandemic, when there is no physical collaboration happening. Maybe not able to go out and get some milk, or not able to go out and create a medium for other. So what these challenges turned into opportunities. So those interviews, we interviewed students and then wrote a sort of nice article about it, how we are sustaining the Cube Lab even in lockdown. Then okay, so here is a thoda sa matlab research based article. I want to just project and then maybe I'll finish my story. So as I am interested in science education research, you study model organisms, and I am interested in studying you, how you study science. So with regard to Cube's work, like how many of you are here working on fruit fly? Come on, raise your hands. I'm not going to ask any question. I just want to know how many are familiar with fruit fly. ठीक है, how many are on working on snail? एक ही आशुतोष और कौन? अच्छा, स्नेल से कोई इंट्रोड्यूस नहीं हुआ है अभी तो. दिस इज द राइट सीजन टू गो एंड फेच सम स्नेल्स. हो गए थोड़े से गोगल गाय आणि और फ्रूट फ्लाई स्नेल अर्थवर्म. ओके ओके, गुड. Any other model system? Anybody working in Pagalophos? Seed germination. Okay. Mosquito. Okay. Anybody on protophore, hydra? Okay. Okay, good. Anybody on nothing? Something. कुछ ना कुछ तो कर रहे हैं. ठीक है. तो like मतलब I want to understand how you understand science. And I think what you're doing with model organisms, you're trying to, it's trying to solve a question. Now, the approach that you're taking is that you are developing some research question, and perhaps you may have your own hypothesis also. So, there is a scientific method that you are trying to follow. When you're asking a question, we need to follow scientific method. So, my interest is that what is this scientific method coming up when you're doing science? So what we did is we asked students to represent, write it in writing. In between the causeries, we asked them, why don't you write what is your experimental design? It is coming up in the causerie. It is coming up, but somewhere that design in your model system that you want to understand. So we got some kind of data around it. ये. Can you see this slide? The other one, the next one. So when there was a workshop, and then students were asking this question. Next slide. Drosophila में this is the question that they were working on. Is there difference in olfactory receptor present in lab and native flies? So what they did is that let us look at the design part. This I wanted to show. There is a petri dish, right? Can anybody explain what is this design? I'm sure you may have done using this method only, because this is sort of standard in Q. Yeah. Can anybody explain this? I mean, chalo. Chalo. Yeah, come on.
No.
हाँ, इधर आके बताओ, एक्सप्लेन वो डायग्राम मैंने क्या बनाया है. वो just flash it. That's like.
Yeah. So as you told, that there are two types of fly that we are looking upon. The one is the very old fly, which has been cultured with that.
No.
The CSB one, and the native fly that we have recently cracked. So the one which has been cultured in lab, it has trapped years ago, I think 60-70 years ago, and from these years it has been cultured in a media that is there already in the bottle. The food is given to it in the bottle, and it doesn't have to search for the food. And the native fly that we have, it has the habit of looking for the food. So we know that they go around food, like find food, by using their olfactory. I mean, like we have nose to smell, they can smell it using some nerves there. So here we have receptors. So here we have, I think this was a agar particulate.
Okay.
And in middle, we will keep some larvae. So fruit fly has 3 steps of larvae, like instas of larvae. First is very small, which is hatch. Then the second insta larvae will start eating a lot, so that it can become larger in size. Third insta larva, and rest will become pupa. So we will use the second insta larva and put around 10 to 20 larvae here, and we will look where the fruit fly is going. So here we have a chemical, isoamyl acetate, which smells like banana. You can also use banana paste here. So the larvae will go till there if it can smell the banana smell, like the essence. And here we have the distilled water as a control to see if like where it is going. So if it can't smell it, it will come here or there, and if it has like the good smelling capacity, it will go here. So we are just checking if the CSBZ, the old fry that was trapped, still has its capacity to smell compared to the native fry that we
Just be on the back button and check is slide visible.
So this what she has explained is an experimental design. It is set up. Ki what are you going to do with that particular petri dish and some larvae. So you have a question. You are trying to find out which of the flies will be attracted to what. Is it smell? And how are you trying to test it with a control? What is your control? A distilled water, right? So that's an experimental setup. So what I am interested in understanding how students understand, what is going around an experimental design. So you need to have control, you need to have a treatment, and then you need to have certain, variables or things that you are studying. So what is the variable here? So variable is like a cause and effect. So in experimentation or experimental design, you need to set up. One is the stat control that you are controlling the variable. So here maybe the treatment, the chemical is under your control because you are varying its concentration and checking, right? And then what you are testing it with is your what are you calculating? The olfactory response index. So here all these things makes an experimental design, at least in fruit fly. So I was studying this with undergraduate students. Now there was another essay on snail assay. So anybody on snail? Yes, please, आप बताइए. Come here. अच्छा, who has, you have? Okay. Acha, हाँ. Okay. So I think Ashutosh, why don't you just tell about the snail essay?
Now,
नहीं वो petri dish का बता दो. Just talk about the petri dish slide, yeah.
Yeah, so in the snail assay,
camera
it's a behavioral assay that we do, and in this we have an arena where we keep the snail at the center, and we are trying to see where the snail moves in terms of two cues that it has. Okay, so over here you will see that at one end we put some particular extract of favorite food of the snail. So let's say cucumber. We create an extract of cucumber and we put 1 drop of cucumber. So there's a circular arena. At one end we put the cucumber extract 1 drop. Diametrically opposite to that, we put a drop of distilled water. Okay. So in terms of olfaction, this particular drop would have a certain smell that would act, we believe, would act as an attractant for the snail because cucumber is one of its food. Okay. So and then at the other end, where we have a drop of water, in terms of olfaction, there is no cue. Now, because the snail has been put in the center, at one end it has a certain cue coming in, at the other end it does not have a cue coming in. And because the cucumber is an attractant, does the snail move towards the cucumber? And then we do many runs on it and to see whether how many number of times does it go towards the cucumber side, how many number of times would it go towards the water side. Okay, and through that we mathematically derive a index called as the olfactory response index. So that is nothing but a mathematical way to express whether it is really going a lot number of times towards the cucumber as compared to this. So you want me to say the further part or just this part, the assay part?
Yeah, maybe conditioning.
Yeah. So then we get results. So students got results that okay, many number of times the snail is going towards the cucumber extract. Okay. Now that we can define as its olfactory behavior. So in terms of cucumber, this is how the snail behaves when it comes to its olfaction, right? And that now that we understand that okay, many number of times the snail is going towards the cucumber, now we can think of conditioning it.
Okay, so now what we do is the cucumber extract that we have, okay, or the cucumber that we have, we lace it with quinine, a chemical that is bitter in taste. So now olfaction is an attractant. The olfactory cue is an attractant, but the gustatory sense, the taste of the cucumber now turns out to be bitter because it has been laced with quinine. And what we do is we give that as a food to the snail, okay, not in the experiment, but otherwise we give it as a food to the snail, and now it picks up that bitter taste. The snail picks up the bitter taste. So it's not a very pleasant experience for the snail. So somewhere we believe that there's some associative learning, that a good smell gets associated with a bitter taste. And now we want to see whether that changes the olfactory behavior. Now it was an attractant, it was going towards the cucumber extract, but this particular unpleasant experience where it associates the good smell, okay, maybe this good smell would always come along with this bitter taste. That associative learning, does that affect the behavior? Okay, and then we again do the experiment and see whether the olfactory response index changes, whether now maybe it was going 7 out of 10 times towards the cucumber when we do 10 runs, does it change to 5 out of 10 times? So has it learned? Has there been some learning that has taken place? And then if there is learning that takes place, if there's a change in the index, how long can it retain this learning? So now we again give it normal food without lacing it with quinine, and how long would that learning stay in the snail? So basically learning and memory studies. Right?
So, so what we were interested in understanding whether students they mentioned experimental design. Okay, control है, treatment है, snail को डाला, वो देखते यहाँ गया है कि वहाँ गया है. But snail में क्या होता है? What we have observed, they miss this conditioning part. The conditioning has to be done separately, and then again the experiment has to be repeated. So somewhere they sort of skip this important part. Why this conditioning has to be done? Because the association has to be checked. So if you see the there is hi-fi mathematics or equations involved. Even though Ashutosh said mathematics and equation and biology में we think अपने को डर लगता है number से, ऐसा नहीं है. So it is like how many times it goes to control and treatment, कुछ minus करो and then upon control and treatment का plus. So it's a very simple mathematics that you have to work out, and then give some kind of you may get desired results or not. Okay, so what is happening here, though the equation is similar, you're sort of understanding the olfaction through fruit fly, understanding the olfaction through snail. So the problem may be a common, but your model system is different. But through different model systems, you're trying to arrive at a common finding or maybe different finding. And another point, I think it's not come till now. So abhi mujhe yaad hai. You may get desired finding, you may not get desired finding. So you celebrate the desired finding. Yeah, like we celebrate goof ups. So that is another very important thing that is happening in Q. If you have done wrong results, wrong in findings, you didn't, the experiment failed, kuch bhi nahi hua, you celebrate. Don't get nervous. So one of the national meets was around this topic, celebrating goof ups. So people, they were, happily coming and presenting. आज मुझे medium मैंने रखा potato, लेकिन कुछ तो गड़बड़ हुआ. The flies didn't get, attracted to the potato. So क्या goof up हुआ? क्यों हुआ? तो celebration of goof up is to understand the problems. Like it's a very common anecdote. Edison ne 1,000 what you say practices के बाद पता चला कि how does a bulb work. But I think he said कि no, I knew how many times the bulb will not work, like 1,000 times. So how many times something will go wrong. So that's why you arrive at science में sort of scientific method which is supposed to be the correct or valid. So you arrive at that by with all these experiences. So it is giving you experiences how to handle organisms, how to take care of your culture, how to take care of the media that you're, how to take care of the readings, etc. So all these are learning experiences, and that's why we sort of celebrate these things. So that is another very important aspect in the CUBE program. I think I think that's what I wanted to share my part of the story. Can you go ahead and एक तो मुझे दिखाई नहीं देता ना. So I wanted to sort of start with an invitation. What is the philosophy behind Cube? What's the Cube model? Like vertical integration, and then participatory base, space no constraint. Anywhere like home lab, kitchen, you can start Cube. It's a collaboration, and you don't need any sophisticated instruments. The local surrounding can become your Cube lab, and then articulating questions, and this meta studio which Nagarjuna has developed and been maintaining, there you have to go and write what you're thinking. So my point that I want to emphasize is that don't just speak, you go out there and write and articulate will help you to think ahead and think better. That is one thing that I want to mention. अच्छा इधर से थोड़ा सा नीचे जाओ, कितना है मुझे, लेट मी जस्ट सी.
35.
That's it. I think you just come here and that's it. Yeah. So with this, and then talking about my interest, my part of the problem, that what I am interested, that understanding experimental design and all, it's a very important thing to learn as a scientific method. So that is my interest in Cube, and I think that's what I wanted to mention as a small story. And welcome to Cube. Thank you. Is Mayur didn't come. He's also
Joining us late.
He's also one of the good old collaborator. I think he'll join.
Veteran incubation.
Veteran. I already feel old now.
Yeah, so thanks, Meena. Thanks for anyone who is new to Cube, and we have certain people over here who are I wouldn't say completely new, but maybe introduced to Cube a year ago, 6 months ago, that way. So an overall view that you provided to us in terms of introducing what this model is and how it works. So thanks for this presentation. So talking about presentations, I think it's about time that we now move towards the actual action that we have for the day. And yeah.
Let the game begin.
Yeah, let the games begin, as Rahul is saying. So we will start with our presentations now.
पेनड्राइव निकाल सकते हैं, मैं अभी निकालती हूँ, हिल जाएगा.
Okay, so then, as has always been the trend that we follow, every session, whether that's a pre-lunch session or a post-lunch session, every session would be chaired by a person. So I would like to call upon it over here for the first session, the pre-lunch session. I would call upon Rahul again to chair the session for the presentations. Okay, so Rahul, would you be there with us? And I would pass on to you the order in which the presentations have been made. Okay, so Rahul, you can keep this with you. And I would invite you to chair the session, and we can, without much ado, we can start with our first presentation.
भी चल रहा है ना.
But then you will have to keep going and changing presentations right there.
ये is that was another PPT which were opening in some other interface. It's loading. Ashutosh, Wi-Fi is not working. Is Wi-Fi not working? Load हो रहा है.
Okay, so
It is
in
only
the meanwhile,
loaded.
we are fixing up some minor technical issues. I would like to call the first group for presenting today, and it happens to be, and I'm very happy it happens to be my favorite model system, which is Drosophila melanogaster, the fruit fly. So I would like to call on the stage the fruit fly group.
So all the fruit fly group, who is all the fruit flies.
Yeah, all the fruit flies.
So maybe we'll keep this trend today. Okay, those who are working on fruit flies will say all the fruit flies come over here. Those who are working on snails will say all the snails can come.
Problem will be with the dogs. We have to ask them if they are. Okay, so everyone who's working on fruit flies in the queue, please come up, and we are starting with fruit flies first. Go ahead. You are the coordinator, right? Okay, so for this group, we have Smriti Gupta as the coordinator, and I'll hand it over to Smriti and the group to talk about how our fruit flies being cultured, they are buzzing and flying in the group. Yeah.
Hello. So as you know that this CLMS is hybrid this time. It is like offline and online both. The very first slide would be like we will start with the online presentation. So it would start with Sonal, who recently started with this fruit fly culture this week, I think 15th of July, and I just dragged her into this. So she would talk about how she got interested in working with fruit flies. So Sonal, are you there?
Yes. Are you able to hear me?
Yeah, tell her that we can hear her.
Yeah, we can hear you.
Okay. So good morning everyone. I am Sonal from DSPMU Rajpur pursuing first year BSc Zoology. The next slide. So we often see some flies in our kitchen attracted towards the fruit. So with curiosity, we had a discussion with our professor and decided to culture these flies, which are called fruit flies. They have brick red color eyes, light color body and pair of wings. Their size is about 3 millimeter in length. Among many species of Drosophila melanogaster is widely used for genetic studies. It belongs to kingdom Animalia, phylum Orthopoda, because of segmented body, class Insecta, order Diptera having 2 wings, family Drosophilidae, genus Drosophila and species Melanogaster. Drosophila model system have received 6 Nobel Prizes till now. Fruit fly got its name Drosophila melanogaster as Droso means dew and Phila means loving. So Drosophila together means dew loving and melanogaster means pigmented stomach. So we found this in fruit flies. And 60% of genes of fruit flies are identical to that of humans. Next slide. Why do we use them as model organisms? So it is found everywhere and it is easy to culture them. It has short life cycle and produce many offspring in this short period of time. And according to Smithy's observation in her home lab, fruit, fruit fly life cycle is about 7 to 9 days. 75% of human disease genes which are conserved in fruit flies so we can use them as a genetic model. We can use fruit flies to study disease like Parkinson's and Alzheimer's. So now when we know about fruit fly and their use in model organism, so now we should know how to culture these fruit flies. So for this I want to call Smithy. Over to Smithy.
Okay, so as already Rahul has explained you all about TRSC media, so that is the media that we use at home lab to culture fruit fly, and even here we are using same media, which I recently made in okay, which we recently made in Cube Lab, which is here. There's a video of how we made it. Can we just please? Okay, so these are the bottles that I think you all have seen yesterday. And okay, so the video is like fast forwarded. Just
I'm seeing it. Wi-Fi connection कहाँ है? It is taking time.
can we have a tea break?
It is taking time. Wi-Fi connection.
Okay. So here you can see we have taken tomato with rava and sugar, which has been boiled in boiling water, and then when it is like cooked. Okay, this is. So then you have to cook it on high flame first, and when it starts boiling, then put it on low flame and let it cook for a while, maybe 1 or 2 minutes. So here I didn't have vinegar, so we took acetic acid, which is 90%, and diluted it, and then added into the media. And this is Vatsal transferring media into the bottles.
Any other video? No.
So now we know how to culture fruit flies. Then what do you think? Like, what would you see after the trap has been made and the culture is there? So the things, the observation which would be taken later on would be explained by Bhagwati ma'am.
हेलो गाइस फ्रॉम छत्तीसगढ़। सो हमें उतनी इंग्लिश नहीं आती है तो मैं अपना प्रेजेंटेशन हिंदी में ही दूंगी। मैंने अपने होम लैब में फ्रूट फ्लाई के लाइफ साइकिल का स्टडी किया है। ये सेटअप मैंने अपने किचन में किया और किचन में जो थिंग्स मेरे पास अवेलेबल थे मैंने उसी थिंग्स का यूज़ किया है। और मैंने ये सेटअप स्टार्ट किया था 20 मार्च 2023 को। इसमें मैंने क्लास लिया था, कांच का क्लास लिया था और उस कांच की क्लास में मैंने टोमेटो का 1/2 पीस लिया था। फर्स्ट डे मैंने ये ऑब्जर्व किया कि फर्स्ट डे में जितने मुझे फ्रूट फ्लाई चाहिए थी उतने मुझे ऑब्जर्व नहीं हुए। फिर मैंने उसी दिन शाम को ऑब्जर्व किया कि धीरे धीरे उनकी संख्या बढ़ रही थी। फिर सेकेंड डे मैंने ऑब्जर्व किया इन मॉर्निंग many fruit flies observed in tomato trees। फिर मैंने उसे ट्रैक किया। सर इंटरेस्टिंग स्टोरी, मुझे समझ में नहीं आ रहा था कि मैं उसे ट्रैक कैसे करूं। फिर मैंने सोचा कि मेरे पास जो अवेलेबल थिंग्स थी किचन में, जैसे हम चाय छानने के लिए एक छन्नी का यूज़ करते हैं, मैंने उसी छन्नी का यूज़ करके फ्रूट फ्लाई को ट्रैप किया। फिर नेक्स्ट डे मैंने उसमें फ्रूट फ्लाई एग्स को ऑब्जर्व किया। Fruit fly eggs in 1 millimeter, and then next day fruit fly eggs convert to larva, first instar larva, and first instar larva is 1 millimeter. Then next day first instar larva growing up and 24 hours later मैंने ऑब्जर्व किया कि जो फ्रूट फ्लाई लार्वा था वो ग्रो कर रहा था एंड मैंने पाया कि यहाँ पे दिख रहा है फर्स्ट इंस्टा लार्वा ग्रोइंग एंड सेकंड इंस्टा लार्वा एंड थर्ड इंस्टा लार्वा देन मैंने पाया कि उसके बाद लार्वा जो है वो प्यूपा में कन्वर्ट हो गया एंड फर्स्ट प्री प्यूपा लार्वा इज वाइट कलर देन मैंने पाया कि धीरे धीरे उसके कलर में चेंजेस आ रहे थे देन इट्स ब्राउन प्यूपा फिर मैंने नेक्स्ट डे देखा कि लेट्स लार्वा और उस लार्वा का जो कलर था वो बहुत ही डार्क हो गया था फिर मैंने इसको ऑब्जर्व किया और पाया कि नेक्स्ट डे जो लेट प्यूपा था वो कन्वर्ट हो गया एडल्ट फ्रूट फ्लाई में
So we have heard about the different Nobel Prizes that fruit fly has got, and now we came to know that the recent Nobel Prize that fruit fly has got, it is in 2017. It is got by Jeffrey C. Hart, Michael Rosbach, and Michael Young for explaining the molecular mechanism that controlling the circadian rhythm in fruit flies. So we all know that the fruit flies are mostly found in daytime. They are more active during daytime and they sleep at they are less active at night. So these are actually self-regulating the circadian rhythm in these fruit flies. So Seema has done the experiments regarding the activity pattern of fruit flies. So Seema will explain that.
Yeah, so hello everyone. Circadian rhythm refers to the specific cycle in the fruit fly. So this specific cycle is very common even in us, like even we are active during the daytime and we feel sleepy during the night time. So similar things were observed in fruit flies as well. So as you can see in this photograph here, so what I did was that we kept a bait like bananas here at a particular place and observed this after a particular time interval and counted the number of fruit flies on the beach. So as I can see in the observation table and the graph, there are two fixed days, one in the morning and one in the evening. So we counted the maximum number of fruit flies that we see in the day. Next slide. Yeah, so I belong to that time in Amethyst Cube, Goa. So there, even my friends studied the same pattern in the year 2022. And as you can see here also in the graph, we observed the maximum fruit flies in the daytime and less active during the night time. Next slide. So here you can see the chatShaala discussion by the Amethyst students with the Cubist. So you provided a great platform. We plotted all the different graphs and came to a conclusion that at what time the fruit flies are more active in Goa, because the experiments were done by the students of Goa here. So we found that fruit flies are more active during the daytime and less active during the night time. So this was the circadian rhythm study done by the Goan Cubist.
I study speed power of native fruit fly and CSBZ flies. That several pages study and experiment the fruit flies. उन्होंने एक कल्चर मीडिया में 70 years तक बॉटल्स में फ्रूट फ्लाई कल्चर किया and many generation mean 70 year में 12 month में 840 month and total generation इसका है 3360 generation. that I study the smell power. दोनों की स्मेल पावर का स्टडी किया गया. उसमें पाया गया कि जो नेटिव फ्रूट फ्लाइज हैं उनका स्मेलिंग पावर देन कंपेयर टू सीएस बीजेड फ्रूट फ्लाइज हाई है, क्योंकि बहुत सालों तक सीएस बीजेड फ्रूट फ्लाइज को बॉटल्स में कल्चर किया जा रहा है, जिसे उनके नेक्स्ट जनरेशन में क्योंकि वो बॉटल के अंदर ही है, इसीलिए उनमें जो ऑल्फैक्शन पावर है वो धीरे धीरे लॉस हो गया है देन कंपेयर टू नेटिव फ्रूट फ्लाइज. इसीलिए नेटिव फ्रूट फ्लाइज में कंपेयर टू सीएस बीजेड फ्रूट फ्लाइज स्मेलिंग पावर हाई है.
Good morning, everyone. This is Swaha from Cube Jalpaiguri. So I was curious to know, like, what are the fruits or vegetables these fruit flies are getting attracted to. So I took a plate from my kitchen and had kept several fruits and vegetables like you can see, like potato, tomato, cucumber, pomegranate, onion, and eggshell. And I have kept these on my study table for 7 days. I have begun this experiment on 18th March 2023 and had concluded on 24th March 2023. So on day 1, I just observed fruit flies on tomato. So I thought they won't be getting attracted to the other food I had kept on that plate. But on the last day, I was astonished to see them getting attracted to eggshell, onion, potato, and cucumber. But the only one who didn't attract any or the fruit fly wasn't interested was pomegranate. The fruit fly, it seemed like the fruit fly wasn't happy to go or suck nectar or juices from that pomegranate. They only wanted things from the other preference which I had provided. So I would give the mic to Smriti to continue. Yeah. So I was confused, like what they like, whether sugar or the eggshell, because they were getting attracted to the eggshell too. So they are kind of attracted to the rotten smell also. And as the fruits and vegetables got overripen or rotten day by day, the number of fruit flies increased. So I observed like 18 fruit flies on tomato, the highest, and 0 on pomegranate. And the maximum number of fruit flies was observed at 12 noon.
So on the very first day, which fruit did you have?
On the very first day, it was tomato.
Okay, so while doing this experiment, we have done many goof-ups, and the first thing that we do is to take evidence. There are many people who culture fruit fly, but they don't take like photos and videos regularly and post them. So that is the first goof-up that we shouldn't do. Secondly, there are people who post it but don't label it. Like bottles are kept, but they haven't been labeled properly. Then we have everything. We have evidence. We took the video, but we didn't post it. There are no updates on the group, and there's no discussion there. Then there are some people who will do the labeling of the bottles, trap it properly. Everything has been collected and posted, but there is no description. Like we don't know what is happening, when it happens, nothing. And last, everything is there, but there are not many people coming to chatShaala. Like there is no discussion. Then how do we proceed further? You won't like discuss, won't get questions, won't be like curious to know what is going on, and it will just stop. So I think we should avoid doing these things and try to do it properly. Like we should be there and carry on with all these things. Like no, we shouldn't stop anywhere. And if you have any problem, just say it. Someone here would have the solution to it. Okay, so here we have presented all things we already have done. There were more few things like at the very beginning. I think Sonal was talking about it, but she was not much audible. She talked about Alzheimer's and Parkinson's mutant that people work on. In the same way, I was thinking to work on one more thing. Like it is not a mutant, but okay. So last year when I was culturing fruit fly, I got this mite infection in it.
And to get rid of mites, I started washing them with a water that was sucrose solution, like 20% sucrose solution. And from there, when I did that, after 2 days, I observed that the larvae that I washed with sucrose solution, it was becoming obese, like fat in structure. So there was this discussion about, can we study diabetes in fruit fly? And that time I was not that sure about it, but later on people started posting different references of how we can study diabetes in fruit fly. So I'm thinking to do something related to it. I don't know how would we do it like exactly, because I'm not that sure about how mutation takes place, or is it a mutation, how do we induce diabetes and stuff. So I'll just say what I'm thinking how it could be done. So first thing, we have to trap flies and culture them, which has already been done. Then we have to make single line cultures. So I have made 3 cultures, out of which in one culture I saw larvae yesterday. The bottles are there in the lab, this lab. Then we have to do identification of the fly when we get next generation fly. Later on, we can try to induce diabetes in them. Now, this is something I don't know much about. I don't know if it is like we will make mutant out of it by using some chemical like mutagens, or some other way, like dipping them into sugar solution again. Then we can, like, when the diabetes has been induced, I was thinking, how do we reverse it, like, again? So that, like, if we can do it in fruit fly, maybe we can do it in humans too. But okay, this is something that I'm thinking, but I don't know how do we go about it. So let's discuss it in chatShaala and move on. And then this is a concept.
You can read from here. You can read from here, from the laptop.
Okay. So here the first thing it talks about is 6 Nobel Prizes. So here it has been listed. I just take a screenshot of this and share it in the group so we all can see it properly. Then there is molecular biology study that we do. Okay, so recently we were discussing about how Tim and Per gene has been studied to talk about circadian rhythm. Like I think Lakshmi was talking about it. So that's how we bring in molecular biology, like when we go into details of it, how genes work and our regulation. And then there is genetics, again related to genes. Then we have biochemistry. So when we would talk about making media, as Rahul was explaining that in lab we use some kind of media, and Samuel Penzer then started making it at his lab, and he didn't know how to make it. So there, when we try to think that what we eat and what energy we get from that, and how we can make same kind of media for fruit fly, so it can get everything that it needs, like carbohydrate, protein, lipids, and all those things. So there, biochemistry comes in. Then we have developmental biology. So as Bhagwati ma'am was explaining about life cycle, there we have developmental biology, and we study like everything, how it becomes like eggs has been laid and it develops into a larvae. It will hatch and larvae would come out. Then the pupa would be formed and then the fly. So here we have developmental biology. Then there is biophysics. So yeah, as we were talking about microscopy thing, like Ashwati did it. Ashwati or Lakshmi? Ashwati. So when they had to do the identification, they took the fly ka wing and they started looking for the vein, like subcostal vein and all those things. So there they used a torch and a good magnifying image was captured by using a phone. So here we have biophysics, like how to take good pictures, good lighting should be done and all those things. Then we have taxonomy. So when we go for identification, we need to know what are the characteristics and where it would belong. So there taxonomy comes in. And then there is concept of evolution. So as again Bhagwati ma'am and later on Meena ma'am was talking about the olfactory assay that we do. So there we are actually checking the CSBZ fly, which has been trapped like 70 years ago, if it still has its olfactory nerves working, or has it been evolved and has become like kind of a vestigial cell which isn't working. So there we have evolution coming in, and there is neurobiology. So as at the very beginning, Sonal was talking about this Alzheimer's and Parkinson disease. These are neurodegenerative disorders, and here we have neurons playing roles. So this is how like one single model system can connect with so many things, and it can give us so much of knowledge that is actually there in our textbook, but it's so like difficult to understand it when we go to read it, because there are so many big complex words. But when you actually try to do it and understand it like practically, then it becomes much easier. So that's how we get context to curriculum. And the last is the collaborators list. So there are many people from different places. So that is me. I stay here in Mumbai. This is Prithvi Raj, sir, who is from Kerala, and he. Okay. So he also worked on food doing the food preference test with Swaha, and again Swaha is from Jalpaiguri, West Bengal. Amrita, she is here, I think. Yeah. So Amrita is also from Aluttur, Kerala, and Seema, the one who was talking about the circadian rhythm experiment that they did. She's from Goa, MHSS. It's a school, college. Then there is Kishan Gupta from GST College, who has recently joined and is planning to work on it and also help us. And then there's Bhagwati ma'am. She is from Chhattisgarh, Pamgarh. Yeah. Shrutika and this that Amrita did, Swaha did, they all were doing together. Then we have Sonal Minch, who has recently joined this week. She's from Ranchi, the first girl who was presenting the first slides. And then we have Lakshmi, who is from SN College, Nadika, Kerala.
So,
Okay, thank you Drosophila group for the amazing presentation. And so we have a few questions. So after every presentation there will be causerie, but I would like to take this opportunity to announce a small tea break here. So and I think it should be small because you can just have tea. How about 10 minutes? 10 minutes is fine, everyone. So 10 minutes tea break, and we'll be back with some questions that also the in-house question, and we have questions from people posting online. So those are very interesting questions that you people have to, tackle. So you need some energy for that. So yes, so Arunan is announcing to sponsor online teas for those who are online. So please enjoy your online teas, and please bill it to Arunan. Okay, we'll meet after the break shortly.
I announce करके just inform and then end it, and then you can tell your business. Just inform करके then you can keep doing it. So you come back to
So, hi everyone on the video button. I hope you can hear me. So, we are taking 10 minutes break for the tea, and you all should go and have a refreshment. So, you can join back. You'll be there logged in. So, we'll start in 10 minutes.
This is what